at the very beginning that the meeting will be recorded, so everybody is aware about this. And if you don't want to be registered, don't show yourself, don't tell anything.
Okay.
Okay, so it's 1 minute before the start, and we can just start to say good morning to all of you that is connected. We will wait just a few seconds more to introduce this cafe. Yes, sure. We are just waiting with our coffee cup.
Francesca, my cup is saying enjoy it.
Enjoy it all, and mine saying don't worry.
So good chance to
trying to make too anxious. Okay, now the time is arrived. I see that a lot of colleagues are connecting now, and thank you very much to all of you. I start introducing myself. I am Francesca Assennato, an environmental engineer and coordinating the area for monitoring and integrated analysis on land degradation and desertification in the ISPRA Institute in Italy. And I have the pleasure today to present this activity I coordinate for the MONALISA project, the Community of Knowledge on Land Degradation and Desertification. And this is, it's a great pleasure to participate in the MONALISA project that is coordinated by Professor Giovanna Seddaio from University of Sassari. And MONALISA is an innovation action funded under the soil mission. Our mission is to design and test innovative solutions to reverse land degradation and desertification, working actively on monitoring and assessment too. And we launch within the Community of Knowledge this initiative of the Café Talk. This is the very first Café Talk we are performing, and so be nice with us with any kind of problem we can encounter. This is the Community of Knowledge that is also shared with another soil mission project on there's a problem with the video, maybe you can help us with the on desertification in the same call of soil mission that is TerraSafe project. And the goal of the community is to bring together scientists and technical experts together with policymakers and representatives of civil society. The community is also planning, in addition to several occasion of Café Talk as today, also opportunities for young researchers, and we will be launching a call for contribution for young researchers soon, this month. So I have to announce, please, to register through MONALISA web to the Community of Knowledge to receive all the information, invitations, and to be actively involved in our activities in the 4-year project MONALISA. I ask if our coordinator Giovanna wants to say hello to this community, and then I will pass the floor to our moderator, that is my colleague Luise, and she will present herself, but we are very glad to have her moderating this first Café Talk of MONALISA Community of Knowledge. So thank you for your attention. Anna, the floor is yours.
Good morning again. this is the first talk I repeat because this for us is an experiment, a very exciting experiment, as Francesca was saying a few minutes ago. I am associated in ISPRA, that is the National Institute for Environmental Protection and Research, and I am the scientific and technical correspondent for Italy for the Convention to Combat Desertification, where I am involved from years. And I am also member of a Team Italy team for G20, G7, UNEA, blah, 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 as expert on land degradation, desertification, and land degradation neutrality. I am not an expert in microbial solutions, also to learn how to pronounce microbial as an exercise for me. But I would try to facilitate and to give a sort of soft guidance to our precious hosts. And we will have a few questions about the microbial-based solution as referred to land degradation desertification to cope with these so huge problems and the challenges and the crisis that is growing all around the world. And I have no special definition that I ask and to our colleague to start with define better and deeply define our topics. Before giving the floor to us so well-known expert, some technical information to permit the audience participation. We will propose a few questions, 3 questions, if I may be more precise, through a Google survey during our conversation. They have a very simple answer that has a binary answer, more or less. And you, the audience, could send us also some questions, only utilize the chat. We will try to prepare 1 or 2 additional questions to the speakers, and they will give the answer if we will have the time available. And now we start with Professor Heulin. Of course, thanks to joining us to find the time in your so crowded agenda. And Thierry is Emeritus Director of Research at the National Councils for Scientific Research in France, CNRS, similar to Italy acronyms, and currently is a member of the French Scientific Committee on Desertification, as well as the scientific and the supervisory board of the Lascaux Cave in Aquitaine. His research focuses on adaptive responses of bacteria to environmental constraint, with particular emphasis on adaptation to desert conditions and the maintenance of biodiversity. His background is in plant biology, microbiology, agronomy, and microbial ecology. And he will start now. We asked him to kindly start with a general introduction to the topic that today we are presenting, focusing on how plants react to drought stress and how microbes may help reducing such stress. Professor Heulin, 7 minutes. Thank you.
Yes, thank you very much for this introduction. And yes, to this question, I would like to just to focus on some points. Desert lands and a very special environment, and the ecology is really special. I've worked in a real desert, but the question is not only the desert, but also the land degradation in Europe. So in deserts, one thing is very special is the biocrust forming by the cyanobacteria. The input of carbon in the ecosystem is not mainly due to the plant, because the plants are very rare, but the biocrust, the cyanobacteria are able to, due to the photosynthesis, to realize the carbon fixation in the system and release some carbon compounds for the microbiota in the desert. Because, as you know, the desert is not really desert for microbiology. You can find a lot of a very large diversity of microbes in the desert. So the first introduction of carbon in the soil in this ecosystem is cyanobacteria, and if we consider the land use in Europe, for example, in Spain, in Italy, or in France, with progressive land degradation, we have to focus on different components of the microbiota, such as the mycorrhizal fungi, the plant growth promoting rhizobacteria. And what I would like to focus on is the role of in the agrosystem in Europe in these conditions of land degradation, is the role of the bacteria able to produce exopolysaccharides in the rhizosphere of plants. And this is true for native plants, for wild plants, but it's also true for crops. All the crops are associated with bacteria able to use the root exudates as a very simple carbon molecules, such as glucose, and these bacteria are able to transform these very simple carbon substrates in macromolecules, and the name of these macromolecules are exopolysaccharides. These exopolysaccharides are able to fix water. With 1 gram of EPS, you can fix 300 grams of water. So around the roots, these bacteria are able to protect the plant from desiccation, and this is one of our main research topic in the lab, because I'm working both in an academic laboratory in Cadarache in France, but also we developed this bacteria in a startup, which is in a spin-off of this academic laboratory, and we mainly focus our activities on this bacteria able to produce exopolysaccharide after inoculation to increase the plant growth and plant health also, and we have some success in this field. So I'm open to questions after this very brief introduction.
Thank you. And we now, could you please focus on a rhizosphere bacteria, better, more technically approach that you can share with our colleagues connected to the conversation? Thank you.
What is the question?
The question is, after this introduction, I asked also before, how plants react to drought stress in a more technical aspect, and how the microbes may help reducing this stress. And then a second part is to focus on a rhizosphere bacteria, specifically, that is your specific expertise.
So, yes. So, as you know, the rhizosphere is a very rich environment in carbon released by the plant. So this is the main mechanism of sequestration of carbon in soil. So if you transform the atmospheric CO2 in photosynthesis after photosynthesis, and about 20% of these photosynthesis are released in the soil under simple molecules such as glucose, what we name the root exudates. These exudates can be transformed by the sum of species, bacterial species. One of them is rhizobium, not the notulating rhizobium, but free-living rhizobium, able to produce a large diversity of exopolysaccharides in the rhizosphere. So these exopolysaccharides are the property of to fix the soil to the roots on the root surface, and to fix the water around the roots. So we have isolated a lot of these rhizobium strains able to produce different types of polysaccharides, and now we are able to inoculate this bacteria to different crops, such as rape, wheat, yes, different maize, and the effect is to decrease the water stress of the plant and the conditions where we can apply this water stress. So if the conditions are very good, maybe the activity of the bacteria is not necessary, but as an insurance, we propose to the farmers to add this bacteria in case of water stress during the plant growth. So this technique is now we use a commercial approach. So we have demonstrated that academic research can be transformed in commercial application with some success.
This will be our last question after the Quirico intervention, because it's a very important observation. Just as very rapid reaction to our first words about the problem of land degradation in Europe. First point is you know very well that France is a few months ago declared itself as affected by desertification, joining the group of the Mediterranean country. We hope that we will be with our group. Yeah. And Francesca has been working a lot on this topic, that is the soil monitoring and resilience law, that the day before yesterday was approved by the Environmental Commission in the approved by the Parliament, the European Parliament. This is the Commission, but is a good step toward the final approval. Thank you, Professor Heulin. We hope that you will reach our community, because these are so specific, and frankly speaking, for me is a new elements that I would add with my fragmented competence on this. Then, Professor Migheli, our second speaker. Wait for Professor Migheli, and then you will have two more questions for both. At Quirico, you are connected.
Yes, I am.
Professor Migheli is a professor of plant pathology at the University of Sassari, fully involved in the MONALISA, because as Francesca was saying, the project is coordinated by the University of Sassari. He is the director of the well-known NRD, that is the Interdepartmental Research Centre on Desertification, and is a member of the steering committee of DesertNet International. And this is a representative of DesertNet Network International in the French Scientific Committee. It is also the coordinator of the global network of dryland research institutions. A lot of things to do, Quirico. Of course, that we share some point, and we've met often. And Professor Migheli research focuses on the biology and epidemiology of phytopathogenic fungi, the increasing severity of emerging plant disease in response to climate change and also to the odor pressure on the environment, their molecular diagnosis, and the plant protection methods. And is fully involved, I would underline, is fully involved, as I said before, in international networking, the scientific cooperation project, and striving to contribute to raising social and political awareness in the field of plant health in the context of the global climate change, as well in the land degradation global crisis. So, Professor Migheli, could you give us your opinion, your expertise in this field in a more articulated way that I was presenting? Thank you.
Thank you, Anna, for this nice presentation. It looks like I'm doing everything, a bit of everything, and eventually nothing in particular. But,
as all of us.
An awful perspective. So basically, no, I'm here because my first love and expertise is also connected with filamentous fungi. So for those who are not fond in soil microbiology, filamentous fungi are tricky microbes that instead of duplicating or dividing themselves like bacteria, they grow as hyphae. So imagine that a hypha is a filament that grows in the apical cell indeterminately, and from time to time it may branches. So you have to imagine this hyphae like branching stochastically, and this leads to a sort of development of a interconnected network of filaments that is known as mycelium, basically. And this indeterminate structure is extremely efficient in exploring and filling empty space, which means that mycelia are well adapted to grow in spatially structured environments such as soils. And in fact, in the forest, for example, they are known to form a sort of web that was named actually Wood Wide Web, WWW, and fungi, filamentous fungi like Trichoderma, for instance, or Mycorrhiza, are able to form this a web of interconnecting plants and other fungi all together, and all the microflora, in fact. So these are a peculiar and important trait of filamentous fungi, because they are like spatial integrators of soil system. They grow and they are able to occupy large volume of the soil matrix, and they are influencing many soil-based services, like those that are underpinning soil ecosystems, basically. The form and the organization of mycelia is very important, because it can be influenced by different factors, like the architecture of the soil, for instance, empty space, etc. The spatial distribution of nutrient resources also, which attracts the growth of filaments, and also the biotic interaction between fungi and other organisms that are present in the soil. And some of the consequences of these spatial organizations of fungi relate to soil structure dynamics, biotic regulation, plant microbial communities, the transport of nutrient elements. So these fungi play a really fundamental role in the health, in the soil health, and in the ecosystem health in general. We tend to forget filamentous fungi or to link filamentous fungi only to moldy fruit, for instance, or moldy plants. This is my field as a plant pathologist, but we should not forget their role in carbon cycling, for instance, etc., etc. Okay. Now, Trichoderma is my favorite fungus because it's kind of is a genus. Trichoderma. It holds many hundreds of species, of different species. It can grow at any latitude. It's a very good saprophyte. You can find the Trichoderma from the desert soil in the Sahara up to the Arctic or Antarctic, and it has many effects, many beneficial effects. And in fact, it is well known as biocontrol agents. This is the main role, which means that it's able to counteract the activity of plant pathogens and to destroy and to use plant pathogens as source of food, of nutrients. So Trichoderma may attack plant pathogens and get nutrients from them, and this reduces the level of pathogenic attacks. But it has also many other functions that are more relevant to this specific Café Talk, which means that it is able, for instance, to produce phytohormones like auxins, cytokinins, and these regulate growth. They regulate, increase the root biomass. It has been shown to increase up to 45% the root biomass. It may enhance water absorption, but it may also enhance solubilization of phosphorus, for instance, because it is able to solubilize phosphorus, some strains. This is very much related to single strains, and lowers also transpiration rate by stomatal control. Some strains are also able to decrease, for instance, sodium build-up in tissues, and therefore it prevents toxicity by sodium, and increase osmoprotectant like proline, and this increased salt tolerance by plants. So plants that are pre-inoculated with these Trichoderma strains, that must be selected, of course. Not all the Trichoderma are beneficial, are able to grow better in saline soils and in the absence, and to cope better with drought stress. Finally, if I may take another 1 minute or two, it is also able to induce systemic resistance in plants through jasmonic acid and salicylic acid pathways that are well-known pathways for induced resistance. It is able to enhance enzymes that are reducing oxidative damage, for instance, in plants, so is an antioxidant boost. And it's also able to reduce hot stress in plant and to modulate expression of stress-related genes in plants. So some Trichoderma were shown to prime plant defense against various types of stressors, both biotic, like plant pathogens, and also abiotic, like drought, saline, stress, heavy metal stress, etc., etc. So it looks like a wonderful creature, and that's why I'm studying it.
Thank you, Professor Micheli. Grazie, Quirico. I learned a lot of things that I didn't imagine in my mind. How rich is the contributors of fungi? We know the fungi, the risotto, no more or less. And we have tied the time to the second round of questions. And we have 2 questions. And this is the same question for both speakers, for Professor Heulin and Professor Migheli. Before the question, please, our colleague Claudia will launch the survey, and we hope that we will be able to send us an answer. It's very easy, very rapid. Did you see the survey? Well,
Okay.
first voting
Launch. Now we
launch.
can go
Right.
on. Okay.
Let's go. So the question is, what is your opinion on the practical exploitation, the practical application of this point of view, the commercial perspectives, the creation of markets, and with a rapid view on the opportunities for the private sector, while respecting scientific achievement and, of course, the environmental impact of this kind of technique. Please, we start with Professor Heulin, that was the first responder. Thanks. Let's go.
Yeah.
Go ahead.
Yes, thank you. So you understand this question is very important for me, because after 40 years of academic research and collaboration on the different private companies, seed companies, fertilizer companies, and the idea was not to wait for something from them, because yes, maybe we have to have the courage to test our ideas and in the field, and this is the reason why we have created this private company 5 years ago. Is a new way for us to transfer this knowledge from the laboratory to the real life, and it's not so easy, but we have to do that to be sure that we have done something at the end of this academic career. And for me, as I mentioned it before, the real motor of sequestration of carbon in the soil, because when you increase this carbon in the soil, you increase the soil fertility and you decrease the land degradation. So increasing the carbon, the organic matter in the soil have both positive effects. The first one is to increase the soil fertility, which is a very good thing for the farmer. But you have also, in a way, a mitigation of the atmospheric CO2. So this is the second point, very important. And the third is to decrease the land degradation. So in this context of desertification and land degradation, the role of this organic matter is very crucial. So sequestration through the photosynthesis of plants is the key factor in this system. So to increase this mechanism, you can have an action on the plant selection. So we have demonstrated that in using pearl millet in Senegal, in Africa, that the different genotypes of pearl millet are not equivalent for the release of carbon in the soil and the formation of this rhizoschist. This is the amount of soil around the roots. So we have demonstrated that the character of the size of this rhizoschist is determined, genetically determined. So this means that the plant selection on this proxy, on this the amount of carbon of soil around the roots can be selected by the seed company. But as you know, at the moment, I have the feeling that seed companies are not on the way to consider this type of plant root character. There are many focus on the resistance to plant pathogen, and so the first for me the first important point is to select the best genotype of plant including the crop. So when we're on the way to demonstrate the same thing on sorgho. So the idea is to select the plant genotype to increase the amount of carbon release in the soil. And the second point is to select the bacterial partner of this plant. And the selection of this bacteria is mainly focused on the production of exopolysaccharides, as I said before. So, considering the both partner, the plant and the microbiota, especially the rhizobiome able to produce these exopolysaccharides, we can have a complete approach to increase the plant resilience in drought conditions. So, coming back to the question, one thing is to have collaboration between when you are a researcher in an academic laboratory. One thing is to have collaboration with private company, and the second way for me, it's very new for me since 5 years, is to have the opportunity and to find the money to create the private company, because we have the end of on all the strategies, and it's not so easy, but it's one way to transfer this academic knowledge to the field. So we are on the way to do that. Maybe different other solutions are possible to make this kind of transfer, but at the end the transfer is necessary because there is some urgence to find solution based on the nature-based solutions, and this is one of them. And we have to consider all the different ways to do that. So I can at the moment the situation for our private company is not so easy, but we are increasing, adding to the main argument for the application of this kind of bacteria is the plant growth. But we are on the way to focus on the carbon sequestration, because this carbon sequestration is a very important point to consider in the future. If
Thank you, Professor. You know that our mantra is conserve, protect, sustainable, manage, and restore land. And this kind of expertise is a very interesting, very useful. I relate this. Thank you very much. And I would also repeat to the audience that we don't open the floor to the question, but only through the chat. And then, Quirico, Professor Migheli, please, some consideration on it, but also focusing on the availability and needs for technical and scientific education training, and the role of to encourage young researcher and practitioner. So, building on this collaboration between a social interest, community interest, and the private performance of the, in the spirit of the public-private partnership. So, Quirico, let's give us your opinion.
My opinion, thank you, Anna. Let me first say that I'm grateful to DesertNet International because becoming a representative in the French Scientific Committee on Desertification, I had the possibility to know Thierry Heulin and also many other colleagues that are following an approach that is multidisciplinary. And this I like very much because you can find sociologists and science policy expert in science policy interface, microbiologists, soil scientists, ecologists, etc., and this really encourage the discussions, and I'm really glad because this opens your mind. I think that it's time to pose the grounds to establish a scientific committee on desertification in Italy as well, as it has been in the past. Okay, I just close this parenthesis to say that trichoderma as a fungus is well known in industry because it is a very strong producer of enzymes, for instance. So all the paper industry, wood industry are based on enzymes that are produced by trichoderma, including tissues, for instance. You know, when we are denim jeans, the decoloration is done by enzymes that are produced by trichoderma. So it's well known, and there is a lot of expertise in liquid and solid fermentation, formulation, preservation, etc., and this encourage its commercial development also in other fields like plant health. Okay, that's why it is so popular in biological control, and now hopefully also as a helper to help plants to cope with stress in drought and saline stress, etc., etc. You ask about capacity building. Yeah, this is crucial, and this relates also to a question that was raised in the chat. It's not just spray and pray. I mean, we have to change the paradigm. It's not enough to inoculate and then hope that something will go on. These are microbials. These are living organisms. So they need specific conditions to grow, to develop, and to do their job. And Thierry has raised a very important point. First, we have to select the crop, the plant, the genotype, and then we incorporate the right microbial to these plants. So it's a sort of personalized medicine, you see, for plants. They need to survive. They need to establish in the soil. They need to establish a relation with the plant rhizosphere. So to do this, you need a lot of knowledge. So that's why it's so important, the capacity building of a new generation of soil scientists that are able to use this nature-based solution in the right way, not just as a chemical compound that or a fertilizer that you add to the plant or to the soil, and you hope that some beneficial effect will come out. Okay, this is my message.
Thank you, Quirico, and I associate ourselves to you in thanking Professor Heulin and thanking you to have a sort of bridge between DesertNet International as a very large organization, and especially between Italy and France. I can underline this our personal backyard, I can say. So, two more soliciting. The first is what I can understand from your speech is that the solution could be tailor-made. Tailor-made because of the differences and the variety of soil types, but also, of course, soil plants needs. And also the necessity to promote an integrated knowledge, a sort of another web of different specialization and the topics. Have you some comments on this? Or we can go maybe presenting some questions raised through the chat, yes? So there is I have two questions. Go directly to this question because it's a necessary, is a coupled with our conversation. And the sustainable monitoring law does not have yet criteria for soil biodiversity health. How could we build these criteria using the contribution of the field of research you are just explained us, showed to us, and going ahead in this your technical scientific and technical suggestion. Beyond inoculating, which are the practices that favor the development of those good, quote unquote, microorganisms? This is a question linking the scientific research to the needs of the governance tools that are essential to guide and to drive this application. Who will respond first, Mr. Heulin? Or I see you are ready to give an answer. Thanks.
Yes, I will answer to the second question. And so I have to stop my phone first. Yes, to the second question, because, and as Quirico mentioned, we have to think about the selection of the inoculum. It can be rhizobium, it can be trichoderma, considering the plant and the soil. So we have to. This is one difficulty of our research, is to integrate the soil, because it's not the same thing to work on the sand in some region in Africa, and in soil with a lot of clays. So we have to consider first the soil and the reservoir of biodiversity in these different soils. They are completely different, and we have to consider the plant. The plant can be a crop, and the plant can be a white plant. So we have to consider also the genotype of this plant, and the selection of the magical bug of the rhizobium of trichoderma is based on these two main considerations. So this is a lot of work, but at the end, if we are thinking about the selection based on this criteria, we can at the end find the best bacteria for inoculation. At the moment, we have something else helping in this selection, is both metagenomics to evaluate the potential of the soil with the different important pathways related to the carbon cycle, the nitrogen cycle. So at the moment, metagenomics is helping us to evaluate the potential of the soil in terms of carbon cycle, nitrogen cycle. And the second part is the selection of the bacteria. We can also we can base our selection on the.
Sorry, please avoid to the microphone open. Sorry, because there is a confusion. Let's go ahead.
Yes. The second point for the selection of the best bacteria to be inoculated is to evaluate their potential using their genome. So we have now very good facilities to get the genome of a lot of strains. So we can have in vitro assays, but we can also have in silico evaluation of the potential of the strains to be sure that the strains are all the important genes involving different mechanisms, but also counter selection, the counter selection of some traits which are not very good for in terms of association with the plant. So you can avoid some pathogens using genome approaches. So these 2 points, metagenomics to make the evaluation of the reservoir of diversity and the genomes of the strain, associated with the evaluation of the soil and the plant genotype, is the best way to select the best inoculum as possible.
Thank you, Mr. Heulin. Once again, we see how relevant is to observe the local circumstances, starting from the soil condition. For us, it's very relevant, and this continuous dialogue between soil and the plants, soil type of vegetation you mentioned, is relevant to be continuously taken into account. And mentioning again the necessity of integrated knowledge, but at local level, linking with the global indication, suggestion, advice that we received from the Professor Migheli. Would you please add something on this point?
Yes, Anna. There was a question. The other question by Mirko Barbero was related to the lack of criteria for assessing soil biodiversity health. And it's indeed a very complicated issue because of this biodiversity is intrinsic to soil health. And as Thierry has mentioned, metagenomics may be a tool, probably not the only one, but I'm quite worried. For instance, there are some drawbacks. I'm getting a bit worried about the fact that what we are going to inoculate in the soil may have deep impacts on soil biodiversity itself. There are several examples. Also in rhizobium, for instance, in the case of rhizobium, it was measured already in the 90s in France that after the application of selected rhizobium strains, the local biodiversity, soil biodiversity in rhizobium species was flattened, was completely competed by selected rhizobium strains that were added to the soil through the seeds, through seed coating. So there may be deep impacts. In fact, one of our best Trichoderma strains belongs to a species that is Trichoderma gamsi. And in some soils of Sardinia, we only found a single strain of Trichoderma in all the soil samples. So which means that this strain is highly competitive and it is able to exclude all the other microflora. So we have to, always not to have a positivistic attitude, but always think about the potential consequences also in this nature-based solution that, nature is cruel. So we may find excellent solution, but they are cruel against biodiversity in the soil.
Thank you, Quirico. We are receiving through the chat some other comments related to the soil biodiversity indicators and the necessity to have an ecosystem approach, considering all the aspects of soil biodiversity. Some colleagues here are working on micro-arthropod communities as regulator and the propagators of soil microbiome. And we would underline what is our opinion about it, the necessity to have consider the relevance of the the rhizosome systems that we are representing in a context of the ecosystem and integrated approach to the soil. Thanks. As a soil assessment tools, soil health assessment tools, of course, because, and I would also to underline that our first question about coming from the audience about the soil monitoring and resilience law about the soil biodiversity in health indicators should be elaborated and kind help us to have a better interpretation of this. Thank you. Have you some comments on it?
Yes, I have just a comment of after what Quirico said before. So this is true. We have, even if what we propose are nature-based solutions, we have to be careful about the eventual effect on the natural microbiota after inoculation. So we can do that, and we have done that using, for example, metagenomics to be sure that after inoculation we have a resilience of the microbiota. So the goal is to increase the population of one strain during the crop, during 1 year, and to be sure that at the end of the crop duration, the microbiota was not affected by this inoculation. So we have now the tools for that, and metagenomics is a very nice tool for that. It was not possible 10 or 4 years ago, 20 years ago. So now it's possible, and it's possible, but we have to do that. And we have to check that inoculation does not disturb the native microbiota. And the example of rhizoblob introduced in France is a very, very bad example, but to my own knowledge is quite exceptional. But Quirico, we are right. We have to check for that, and we are able to do that. So this is a very important point.
Thank you, Professor Heulin. Very nice to. Thanks again for participating to our first Café Talk. Let you ready for other conversation. And today we learn once again how complex is the life on the soil and how complex is interaction with our health, how the health of the soil is it's necessary for our quality of life, our well-being at all levels, health as a personal sanitary health, but also to our social and economic benefit, because it's connected in the discussion. We have a lot of projects also on this, on degraded or restoring degraded areas, also for economic and social benefit. So thanks again, and I give the floor to Francesca for the final salutation to us. Thank you, Francesca.
Thank you very much, Anna, and thank you all for being here. Just two very short comments about all the knowledge that we heard and hopefully exchanged today, also in the future. The first is the importance of these approaches to DNA that is also underlined in the soil monitoring law discussion in an integrated way with other indicators. And so it's one point that we confirmed today with this conversation. The second is really important to hear by Professor Heulin in particular. So about the need to transfer researchers, are not only the research results to the market and to the in-field activities. And this is food for thought about how to help and guide young researchers in their future professional life, not only academic. And maybe it's time to dark a little bit our hands, going outside books and videos. My personal thanks to everybody, to the MONALISA project and the ISPRA staff, that plenty of
ladies. There's a group of ladies.
I want to show to all of you, also the colleagues helping us in doing things today. It's a cooperation. Every day is a fight against technology, let me say. But thank you very much. This was the first MONALISA Café Talk.
But not the last.
Not the last. The next one will be in October, and we will launch soon the call for young researchers on the topic of this new Café Talk that will be on machine learning, remote sensing, and artificial intelligence applied to land degradation and desertification. So thanks a lot to everybody. The video will be available through the MONALISA website. So thank you very much to all of you. Bye-bye. See you in October.
Thank you so much. Bye.